Hello everyone. Today we will be talking about microbiology techniques, and the question came in the year GATE Xl-S, and the question number is question number 19. Okay, and the question states that a 100 microliter alicot 10 to the power minus 4 dilution of the bacterial culture plated on the nutrient agar gave 4 colonies. The bacterial stock concentration in million cells per ml rounded off to 1 decimal place is. So what the question is asking is that it is actually asking the concentration of the bacteria in the stock solution. So there is a technique by which we can actually calculate that by doing serial dilutions. So this question has been asked from that topic. So first let us get accustomed to that technique. Okay. So the main idea behind this is that we need to count the colony forming unit per ml in the stock solution. Okay, in this question. So what is colony forming unit? It is a measure of viable bacterial or fungal cells. Okay, so it may be like that in your stock solution, some cells are living, or whereas some cells have died. Means the living cells are the viable cells, and the colony forming unit is an measure of those viable cells in your stock solutions of bacterial cells or fungal cells. Okay, so in direct microscopic counts, cell counting using hemocytometer, where all cells, dead and living, are counted, but CFU measures only viable cells. Means in you have to count the cells. How do you do that? Or the colonies, how you count those colonies? You actually take help of hemocytometer and try to count how many colonies are present. So from there you can actually get an idea how many cells are present or whether they are dead or living. For that you can use some kind of dye to detect the whether your cells are dead or living, like DAPI staining you can do, and you will be able to know whether your cells are living or dead. Okay, and but what is important in colony forming unit, we are more concerned about those cells which are viable, which are living cells. We will only count those. Okay, so the number of bacteria. So another way to look into is this is like the number of bacteria in 1 ml of the original sample. So if someone asks you to calculate the number of bacteria in 1 ml of the original sample, then that can be defined as the CFU per ml. Okay. So CFU of the original sample per ml is this. It can be also written as the number of bacteria or fungi in 1 ml of the original sample. So what basically you do is that you have a stock solution. So from that stock solution, you actually take equal amount of volume and add to subsequent volumes so that you dilute your concentrated the stock solution by a equal dilution factor. Means equal dilution, not equal dilution factor, equal dilution. Means if you are diluting your sample tenfold, then you have to do this tenfold dilution for 3 to 4 steps. Okay, for example, here you are adding 1 ml to 9 ml culture. Again you mix this, shake it properly and take 1 ml from here and add into another 9 ml solution, or 9 ml water or buffer or LB media. Okay, so in this way if you do, so how many times are you diluting your sample? That will be actually giving you the idea of the dilution factor. And what is the CFU per ml formula? That is number of colonies. So from here, for example, from here you take means the bacterial culture and actually plate it and get this kind of colonies, okay, in the bacterial plate, and then count the number of colonies. So that will be your number of colonies and multiplied by total dilution factor. So how you can calculate the total dilution factor, I will come in the next slide. Divided by the volume of the culture plated in ml. So how much volume of the culture you have plated in ml, that you need to consider so that you can get the CFU per ml. Okay. Now what you need to see is how to calculate this dilution factor. So what is this dilution factor? Dilution factor or dilution ratio is used to express how much the original stock solution is present in the total solution after dilution. Or dilution factor is the ratio between the final volume and the initial volume of the solution. So dilution factor is an idea that how much your stock solution has got diluted in this serial dilution process. Okay, for example, here it is getting tenfold diluted. Okay, 10 times diluted it is becoming. So what is the dilution factor? Dilution factor is 1 by dilution. So dilution is basically the initial volume divided by the final volume. So the initial volume that you have taken is 1 ml, and the final volume that will happen is 1 plus 9, that is 10 ml. So what is your dilution here is 1 by 10. And what is your dilution factor? That is the reciprocal of dilution, that is 1 divided by zero point 0.1, that is 10. Okay, so your dilution factor is here 10. So this is the way you can actually calculate your dilution factor and put it into this CFU per ml formula to get the CFU per ml or the number of bacteria in 1 ml of the original sample. Okay, so by looking into this, now let us look back into the question. It has said that it has made 100 microliter alicot of bacterial culture, and they have plated it on the nutrient agar, which gave number of colonies is given as 4 number of colonies, and the dilution is given as 10 to the power minus 4, and 100 microliter has been plated, they have said. So the bacterial stock concentration, so what is the bacterial stock concentration they are asking. So for example, if you consider that, so they are asking what is the concentration of this bacterial stock solution, and they have what they have given, they have given the number of colonies, they have given the dilution, and also the how much volume they have plated in the bacterial plate. Now let us try to solve this question by remembering the theory that I have just talked about. So what is given? The alicot with 10 to the power 4 minus 4 dilution is 100 microliter. Means 100 microliter is your final volume that you have plated, or that you have taken the alicot, which means 0.1 ml. Okay. Now we know what is the formula of colony forming unit. So that is the number of cells multiplied by dilution factor divided by volume. So number of cells is given as 4, or number of colonies here is given as 4, right? So it is 4. Okay, so here we are considering one colony is equals to one cell. Okay, so approximately, that is not like that, but we are for approximately we are considering that. Then dilution factor is 1 by dilution. Okay, so dilution is given 10 to the power minus 4, so your dilution factor is 10 to the power 4. What is the volume they have given? That is zero point 0.1 ml. So in 0.1 ml, how many cells are present? Okay, so these 4 cells are present. So they have said you that 4 colonies are present. Okay, so culture plate on the nutrient agar gave 4 colonies. So how many cells are actually present in the 100 microliter or 0.1 ml that we have calculated? That is 4 into 4 multiplied by the 10 to the power 4, because the dilution factor is here 10 to the power 4. Okay, so in stock solution of 1 liter or 1000 ml, okay, so how many cells should be present? Because we are interested in that in 1 liter or in 1000 ml, how many cells are present. Okay, so there we have calculated like that 1000 ml means in 0.1 ml if 4 into 10 to the 4 cells are present, then in 0.1 ml how many cells will be present? That is 0.4 into 10 to the 4 divided by 0.1 in 1 ml, and in 1000 ml what will be the amount of cells that will be present? That is 4 million cells. So how you are getting this 1 liter volume? You are getting this 1 liter volume because you have 0.1 ml of the final volume. Okay, and the dilution factor here is 10 to the power 4. So how much you have diluted from the original sample, from there you can get the total volume. So zero point 0.1 into 10 to the power 4, that is 1000 ml. So originally your stock solution is of 1000 ml. Okay, so I am reverse calculating that. So that's why I have found out that in stock solution, where 1000 ml of media was present, how many million cells were present. Okay. So from here, we have come to know that in 1 ml, how many cells will be present. Similarly, by unitary method. So by 1000, you divide it, it will be 0.4 million cells. Okay. So in 1 ml, zero point 0.4 million cells you will be actually getting. So the answer to this question will be zero point 0.4. For your reference, you can actually go through these two courses in NPTEL, that is Advanced Microbiology by Arup Kumar Mitra and Microbiology by Dr. Gurvinder Kaur Saini.